Hi, it is my pleasure to introduce today's speaker, Dr. Patrick Murphy from the University of Rochester, Department of Biological Genetics and the Department of Biology Research. Dr. Murphy received his bachelor's degree from St. John's University and then worked as a training fellow at the NIH. He went on to obtain his PhD in genetics and development from Cornell, studying mechanisms of epigenetic regulation. He completed his postdoctoral training at the Howard Hughes Medical Institute, and he has been an invited speaker at numerous universities and conferences. He currently studies epigenetics and regulation of transcription in development and disease, and we are very excited that he is here today. So thank you so much for coming to speak with us, Dr. Murphy.
Great, thank you. It's really my pleasure to be here to tell you about my research, and I really very much appreciate the invitation. So today I'm going to tell you stories about it's kind of a 2.5-part story. And it begins with the work I did as a postdoc at the University of Utah, and then it kind of ends with the work I did in my own lab recently, and then some follow-up studies at the University of Rochester. And generally, the connection in this research is the idea of chromatin packaging and gene regulation, but we'll be talking both about development and potentially a little bit about disease. There's some projects in my lab ongoing with disease, and I'll hint at them at the end, but they might not have made it into the talk completely. Okay, so like I said, postdoc work at the Huntsman Cancer Institute at the University of Utah, and now my current work is at the University of Rochester, and I've been an assistant professor since the fall of 2018, so just about 2.5 years. So the work I'll tell you about from the University of Utah involves my studies using the Zebrafish model and how we defined these placeholder nucleosomes, which contain the histone variant H2AZ, and how this process regulates epigenetic reprogramming during stem cell formation. And then after that, I'll tell you about how one of the factors that regulates H2AZ, called AMP32E, controls gene activation in mouse cell culture, and we really are interested in mechanisms of transcription activation here. And then finally, we'll kind of come full circle back to the zebrafish with some recent studies in my lab investigating how these same molecular mechanisms that we uncovered in mouse might be contributing to developmental phenotypes in the zebrafish. Okay, so I want to start with some background, and I always think it's important to talk about what epigenetics actually is. And that's because it is one of these buzzwords that everyone uses and has kind of eroded into nonsense. So if you like go on Amazon and you type in epigenetics, you'll find all this crazy stuff, like face cream and shampoo and even pet food that all say epigenetics on them. So we don't really, as a society, understand what epigenetics is. So I feel like as scientists, it's important for us to kind of continually remind each other in our echo chamber of what epigenetics is. And so back in 2008, one of the real leaders and early pioneers in the epigenetics field, Adrian Bird, was quoted in this cool article in Nature where he said, epigenetics is a useful word if you don't know what's going on. And if you do, you use something else. And that's true, actually. I go to the Epigenetics Gordon conference every 2 years, and most of the people there don't even use the word epigenetics because it's become something else that it wasn't really originally intended to mean. So what does epigenetics mean? Well, it was coined by Conrad Waddington in 1942, and Conrad Waddington was really a developmental biologist, and he was bringing together the fields of developmental biology and genetics in a way to kind of understand how genes can function in the context of a phenotype in an organism. And so he said that interactions between genes and their products, which bring about the phenotype, is really what epigenetics is. And in simple terms, we can kind of think about this as gene expression regulates cell state. So interactions between genes and their products is what I kind of think about as gene expression, which bring about is simply a way of saying that they regulate. And phenotype at the cellular level is really what the cellular program is, or the cell state. So what do I mean? Well, if you think about two defined cell types, here the example I have is a neuron and a macrophage. And although they have very distinct function, they really have identical DNA sequences. And it's the gene expression in the macrophage and the neuron which define the identity of those cells. And different genes are active in different cell types. So here I've got an example with this Venn diagram where in the macrophage, specific genes are on and specific genes are off. And in the neuron, the gene which might be off in the macrophage would be on in the neuron, and the macrophage specific gene would be off in the neuron. So there's also a very specific cell type called a stem cell, which has this defined characteristic called pluripotency, which means it has the capacity to differentiate into all embryonic cell lineages or cell types. And this cell type is particularly interesting to me. It's the cell type I kind of began working on as a graduate student, and I continued working on as a postdoc in the Zebrafish model. And it'll be the focus of a lot of the talk today. So what, in terms of epigenetics, defines stem cells? So if a macrophage is defined by a particular gene expression program, and the same for a neuron, well, what defines the cell type that doesn't really have a gene expression, program? It's not a differentiated cell. Well, it turns out there's this really specific epigenetic state in stem cells called poised chromatin, where a gene that needs to be turned on later during development is held in this poised state, kind of somewhere between on and somewhere between off, so that later on during development, that gene can be turned on, or that gene might remain silent if that gene isn't necessarily needed in that particular cell type. So now we've kind of discussed stem cells, and I've introduced this term chromatin. So I'd like to focus a little bit more on what chromatin is. So chromatin is simply thought of as how DNA is packaged in the nucleus. And we've all kind of seen this example where DNA is wrapped around the nucleosomes, forming this beads on a string model. And then compaction occurs further where looping, and eventually you get these mitotic chromosomes where there's a high degree of chromatin packaging. So chromatin is really the interaction between DNA and proteins so that they can form this compact structure. And at the simplest resolution, nucleosomes are the finest, resolution of a chromatin molecule. So not only is chromatin important for controlling compaction, but it's also important for controlling gene expression. And so most of the chromatin in your genome can be defined as either open or closed. And defining chromatin in this manner partitions DNA into two basic classes, where gene promoters and enhancers, replication origins, and insulator sites are generally in this open conformation. And closed chromatin occurs at gene bodies, intergenic regions, repetitive elements, and telomeres and centromeres. So the general idea is that wherever genes need to be turned on, wherever transcription needs to occur, wherever transcription factors need to bind, this is generally open chromatin. And wherever you want to prevent all that stuff from happening, that's closed chromatin. So just to make this point crystal clear, imagine that there's a transcription start site where a gene turns on, and it's located at this particular location on the DNA. And that if that piece of DNA is wrapped around the nucleosome, now that might prevent the transcription machinery from binding that piece of DNA. And so that gene is off because the chromatin is closed. But if the DNA is unwrapped or loose in a way that we define as open, now the transcription machinery can access that portion of DNA and the gene might be turned on. So really this is open and closed chromatin. It's a concept I'll come back to throughout the talk. On top of this, chromatin can also be regulated in the form of other epigenetic marks. And the example I have here are histone modifications. And so the histones are the proteins that make up the nucleosome, and they can be modified on their tails, with post-translational modifications, either methylation, acetylation, phosphorylation, and these can occur at a variety of amino acid residues. So there's been numerous studies investigating the molecular function of these modifications, and we've kind of assigned, at least broadly assigned, function to each of them. Some are involved in activation of gene expression, like H3K4 trimethylation, and some are involved in gene silencing, like H3K27 trimethylation. And it's really the location of these marks which contributes to the opening or closing of chromatin, as well as the ability to recruit activating or silencing machinery in the genome. Another form of epigenetic mark or chromatin feature is histone variants. So the histone proteins that make up the nucleosomes are either can come in the canonical form, which occurs in the vast majority of nucleosomes in the genome, or they can come in this variant form, where specific amino acid substitutions or variation occur across this variant histone. And these differences in the two histone flavors, I guess you can say, gives the histone and the nucleosome specific function. So this particular histone variant I'm showing you here, H2AZ, is the one that my lab focuses a lot on, and it also is known to occur at more open chromatin regions where gene regulation occurs. Finally, I'll talk today about DNA methylation. So this occurs when a methyl group is added to the 5th carbon on the cytosine ring, and it happens in a CG dinucleotide context. So the methyl group can either be added on the top strand or on the bottom strand, and it kind of forms this palindromic sequence here. Finally, high levels of DNA methylation are associated with gene silencing, and low levels of DNA methylation are associated with gene activation. And on here, I'm going to use this kind of cartoon to represent high levels of DNA methylation, these filled in circles, and low levels of methylation with these open circles. All right, so back to our earlier question. So how is the genome of a pluripotent cell defined? And I talked about poised chromatin. Well, it turns out there's a really special type of chromatin that exists at the promoters for these poised genes. This chromatin has an activating mark, H3K4 trimethylation, at the same location as a silencing mark, H3K27 trimethylation. And the idea is that these marks antagonize one another and they hold the gene in a poised state, so that later on during development, all you would need to do is remove one of these marks so that the gene can be stably silenced if you remove H3K4 trimethylation, or it can be stably activated if you remove H3K27 trimethylation. So this kind of brings me to the questions we started asking when I was a postdoc, namely, how is poised chromatin established during development? So if this very important chromatin signature underlies the programming of a stem cell, we wanted to know, well, how is it that this programming occurs? How is it that the epigenetic marks know where to go so that they can form a stem cell during the earliest stages in development? All right, so before I kind of get into the experiments, I want to just put in a plug for Zebrafish. So we addressed this question in Zebrafish because of the huge advantages that the Zebrafish model provides us. And so I'm going to give you a quick comparison. So in Zebrafish, gametes and embryos can be collected without sacrificing adults, right? So external fertilization, it's great. You can just harvest embryos off the bottom of a petri dish. Similarly, you can collect sperm and eggs without having to sacrifice the adult. A single mating pair can generate 50 to 200 embryos at a time, whereas in mice you might get 6 to 12 embryos in a litter if you're lucky. At the stage when the zygotic genome becomes active in zebrafish, there are 4,000 stem cells, whereas that similar stage in mouse, there's only 4 to 8 stem cells in the embryo. And finally, generating these stem cells over a developmental time course takes only 4 hours in zebrafish compared to 36 to 48 hours in mouse. So the take-home message here is that zebrafish are like yeast. Like they can just give you truckloads of stem cells almost on demand. And then you can take these stem cells and do somewhat complicated genomics experiments or biochemistry experiments that you wouldn't otherwise be able to do in vertebrate systems. So I'll tell you a little bit more about early zebrafish development and the establishment of pluripotency. So just like in mammals, sperm and egg come together to form the zygote, except in zebrafish, the zygote undergoes 10 cell cycles of just rapid mitosis before the stem cells form. And this allows for the many, many, many numbers of cells that we can use for our studies. So to investigate this question about how poised chromatin is established, we needed to first ask two questions. When during development does this occur? And where in the genome do bivalent chromatin marks arise? So I'll just summarize some work that had been done previously. So we kind of had an idea for where to start. So we knew that in eggs, DNA methylation marked a lot of the genes that went on to become bivalent poised chromatin. And in sperm, although these genes were also silent, there was low levels of DNA methylation at the promoters for these bivalent genes. And the histone modifications were already present in sperm. So the simplest explanation for how is poised chromatin established is really just that sperm passes on the right epigenetic marks to the embryo. Nothing else happens. Really simple. Mom doesn't matter. But, and that was generally the idea for quite a while. I think in 2008, they made this discovery in many organisms, and it was just generally assumed that throughout this process, all you needed to do was maintain those epigenetic marks. But then we asked, well, really what's going on in this early stages of development? Is it really maintained? And in a series of studies kind of around 2010 to recently in humans, just I think 2 years ago, they confirmed this result now in humans. Turns out the epigenetic marks are lost from sperm to the embryo. And although DNA methylation is still lacking, the histone modifications that are normally present at these bivalent marks are erased immediately after fertilization during this cleavage phase of development. So with that, we were kind of puzzled. Well, how is it that bivalent chromatin knows where in the genome to be established, and at what stage does this happen? So this is kind of the state of things when I was working on this project in my postdoc, and to kind of get an idea for how this worked, we developed this kind of unifying hypothesis about placeholder nucleosomes. Here, the idea is that these placeholder nucleosomes may also be inherited from sperm in kind of a generic bland flavor, but they may block the DNA methylation enzymes to maintain low levels of DNA methylation, kind of preventing accumulation of DNA methylation. They might be passed from sperm to embryo, explaining why the sperm DNA looks the same as the embryonic DNA. And then they would have to be installed at the correct locations on the maternal genome in order for the maternal genome to be converted from an egg to a stem cell. And then finally, these kind of bland placeholder nucleosomes would almost act as a platform for the establishment of bivalent chromatin. So in this way, the single nucleosome that kind of resides at particular sites would accomplish all of these goals. So then we went kind of hunting for this idea, this conceptual placeholder nucleosome. And before I get into how we eventually found it, I want to quickly go through some of the methods we used. For those of you who are not so familiar with these genomics methods, I'm sure many of you are. One of the techniques we used is chromatin immunoprecipitation and sequencing. And so the very simple way to explain this is you take a tube that contains chromatin. Some of that chromatin has an epigenetic mark you're interested in. There's many pieces of chromatin that have all the stuff you're not interested in. Using an antibody, you selectively purify out the chromatin that you're interested in. You extract DNA and sequence that DNA. When you align all the reads across the genome, you can find where DNA is enriched at particular sites, and these locations correspond with the epigenetic mark location in the genome. That's essentially how ChIP-seq works. And when we measure those locations in the genome, you'll either see it like these kinds of enrichment plots where the x-axis is genome coordinates, or you'll also see these kinds of heat maps where pieces of the genome have been chopped out and we're measuring the enrichment on a colorimetric scale, and then we can stack different portions of the genome on top of one another in an ordered way, and this will end up being these kinds of heat maps where you'll see the center will be the center of the particular slice, and then the red color will be how much enrichment there is emanating from that center point. So you'll see a lot of heat maps like this, and here the important thing to remember is that the rows correspond to different slices of the genome that we've kind of chopped out. Finally, I'll talk about how we measure DNA methylation using this technique called bisulfite sequencing. I'm not going to really get into the biochemistry of this. I'll simply just say that we can measure the percent DNA methylation in a population of cells kind of as either 100%, 0%, or any percentage in between across the entire genome at single CpG resolution. So that's called bisulfite sequencing. And again, you'll see it as kind of these like bar plots rather than a smooth enrichment plot like a ChIP-seq experiment. Okay, so when we were beginning with this, what is the placeholder nucleosome? The first place we decided to look was the sperm chromatin. And there were a number of different epigenetic marks we knew from the literature might be part of the placeholder nucleosome. So we initially did ChIP-seq, looking at all of these different histone modifications and DNA methylation. And we knew the Hox genes were a cluster of genes that are bivalent in the embryo. What we found was that many of these resided at the same location where there was low levels of DNA methylation, but some of these marks were not quite as abundant. So that narrowed our focus to some of these, and these two from previous studies we knew were not maintained from sperm to embryo. So we really focused on this histone, which is H2AZ, and in fish it's called H2AFV. So I'll kind of use the two interchangeably. I have kind of a nomenclature here. And then H3K4 monomethylation is the methyl group added to the fourth lysine on histone 3. We looked across the entire genome, and what we found was that DNA methylation was anti-correlated with the monomethylation, the trimethylation, and the H2AZ mark, where it was not nearly as strongly anti-correlated with the other epigenetic marks. So then we did this same experiment looking at different stages in zebrafish development, both prior to the establishment of stem cells and after the establishment of stem cells. So this establishment occurs at the time of zygotic genome activation. That's what ZGA stands for here. And so the hope was that we would identify a cluster of epigenetic marks that were all correlated with one another and anti-correlated with DNA methylation. And these epigenetic marks might represent that placeholder nucleosome. So here, the H2AV, again, H2AZ, and H3K4 monomethylation stood out. So next we needed to just investigate mechanistically whether this was involved in reprogramming the DNA methylation, because this is an important component of our model. So to investigate this, we decided to do a genetics experiment to manipulate H2AZ localization in the genome. And rather than manipulating H2AZ itself, we decided to reorganize H2AZ by manipulating the enzyme that is involved in installing H2AZ. Here our hypothesis was, if we prevent H2AZ from being installed, then we would expect regions that normally have low levels of DNA methylation would inappropriately gain DNA methylation. So we did this, we did bisulfite sequencing, and we found indeed when we injected a morpholino against SR-cap in order to decrease SR-cap, we saw that there was an abundant increase in DNA methylation throughout the genome. We also confirmed the phenotype of these morphins using a genetic null, but I don't really have time to get into that today. Next, we wanted to kind of do the opposite experiment where we wanted to manipulate removal of H2AZ. So during this whole reprogramming process, the egg needs to remove H2AZ from genes involved in egg programming so that it can convert those genes to a stem cell state where that gene might be off. So in order to go from high levels of DNA methylation to low levels of DNA methylation, we hypothesized that H2AZ must be removed from those sites. So if we prevent this removal by getting rid of ANP32E, we would see inappropriate maintenance of H2AZ. And based on our hypothesis, then we would expect inappropriate loss of DNA methylation at these sites. So for that, we did a genomics experiment where we first measured H2AZ changes in the ANP32E mutant sperm and in the embryos where there was both maternal and paternal loss of ANP32E. What we found was that there was an abundant accumulation of H2AZ, both at sperm-specific sites and at sites that were where H2AZ accumulated both in sperm and in the embryo. We defined these as differential variant regions, meaning there was differences in H2AZ enrichment. We then looked at single CpG dinucleotides throughout the genome, and here I'm just showing you the sperm DNA methylation compared to the wild type compared to the mutant, and a reduction in DNA methylation would show up in this lower corner here, and 87% of the changing DNA methylation we saw occurred in the decreased direction in sperm and 67% in the embryo. So this was a strong indication to us in combination with our earlier experiments that indeed H2AZ was restricting DNA methylation patterns. All right, so finally, I'll just kind of summarize many other experiments I didn't have time to tell you about, but what we found was that these placeholder nucleosomes are present in sperm. They're maintained at the locations where stem cells activate genes, at housekeeping genes involved in normal cellular processes. They're also maintained at the genes involved in early embryonic development so that bivalent chromatin can be established and hypo-DNA methylation occurs at these regions. At the genes involved in later stages of development, placeholder nucleosomes did not exist, and there was antagonism of these nucleosomes by ANP32E. And reprogramming of the maternal genome occurred in a manner that placeholder nucleosomes were established at early development genes from egg DNA, and they were antagonized at the genes where they were involved in later stages of development. Okay, so now I started my own lab in 2018, and we started asking somewhat similar but different questions. The question I was most interested in when I started my lab was, well, how do placeholder nucleosomes regulate gene activation? One of the things I didn't have time to tell you about, but I'll tell you now, is in these studies, we found that the locations where these placeholder nucleosomes accumulated the most coincided with this high level of gene activation. So I'm showing you ChIP-seq at the top and then RNA-seq at the bottom. And there was a strong correlation between increased H2AZ and increased gene expression. And it really wasn't clear to us how these placeholder nucleosomes were regulating this process. So here I kind of developed this hypothesis that maybe these placeholder nucleosomes have some role in this opening and closing of chromatin. And if the placeholder nucleosomes facilitate opening chromatin, then you would have to remove them in order for you to get closed chromatin, and vice versa. And also have to install placeholder nucleosomes to get open chromatin. So we thought about this and we thought, well, how does opening and closing of chromatin occur normally? What's known about this process? Well, it involves these specialized epigenetic complexes called nucleosome remodelers. And these remodelers are many proteins that form these kind of macrostructures that can then bind to DNA and slide or eject nucleosomes so that places which would normally not be accessible to the transcription machinery can now become accessible to the transcription machinery. So it's really through this nucleosome remodeler action that you can go from a closed chromatin site to an open chromatin site. So with that, we kind of built on our hypothesis and said, well, maybe these placeholder nucleosomes are somehow facilitating the remodeler in order to activate genes. And in this manner, ANP32E might actually be involved in preventing H2AZ incorporation and actually help with keeping chromatin closed. So this was the hypothesis that I used to kind of start my lab. Again, we have another technique here. So I'll tell you about how we measured this. We measured this using a technique called ataxic, which stands for the assay for transposase accessible chromatin, ataxic. And this relies on the ability of TN5 transposase to get incorporated into the genome. And when TN5 is incorporated, it drops off these little PCR primer sequences and adapter sequences for high throughput sequencing. And after it drops these sequences off, it leaves behind these tags in the genome. And it can only drop off these PCR tags at locations that are accessible, and locations that are inaccessible, the transposase cannot enter. So after you've applied the transposase to the chromatin sample, you can simply PCR amplify your DNA and get a pretty good idea for where the genome is open and where the genome is closed. So we used this method and we compared it with H2AZ localization, both in mouse stem cells and in mouse fibroblasts. And what we found was that H2AZ enrichment sites corresponded really strongly with sites of open chromatin. When we looked across the entire genome, we found that sites where H2AZ occurred were almost always sites where there was high levels of chromatin accessibility, and many of these genes were also active. So I'm showing you RNA here where you can see that there's high levels of RNA transcripts where the H2AZ sites are, and low levels of transcripts where there's a lack of H2AZ and a lack of chromatin accessibility. Then we asked, well, what happens when H2AZ expands in the mutants where we get rid of ANP32E? So we looked at chromatin accessibility, and it's pretty clear here there's a general increase in chromatin accessibility across all these accessible sites in the genome. We performed differential accessibility analysis, and we found that the majority of the genome was increasing accessibility in the absence of ANP32E. Then we said, okay, well, if ANP32E is regulating H2AZ, we should be able to rescue this by simultaneously removing or depleting H2AZ. So we used an siRNA to knock down H2AZ, and then we repeated these experiments. And sure enough, we saw that there was a loss of accessibility specifically over the sites where H2AZ resided. And when we measured H2AZ marked sites versus unmarked H2AZ sites, we found that they increased in the absence of ANP32E, and then they decreased when we further removed H2AZ. And we did not see that same trend when we looked at sites that were not marked by H2AZ. So this kind of led us to say, yes, it does seem like ANP32E might be regulating chromatin accessibility, but what about the remodeler? Is that what's going on with this remodeler? Based on our hypothesis, we expect to see differences in that remodeler. So here we performed a technique called cut and tag. It's a newer method similar to ChIP-seq, where we mapped SMARCA4, which is the predominant euchromatic promoter remodeler in vertebrates. We found that many of the sites where H2AZ resides also have SMARCA4. Nucleosome positioning does tend to increase when we get rid of ANP32E, indicating that the remodeler is indeed more active. And when we looked at the remodeler itself, we found that the remodeler increased when we got rid of ANP32E at sites where accessibility increased, and at sites where accessibility decreased, the remodeler also decreased. And so based on this, we developed this model where H2AZ dosage is really what's controlling nucleosome accessibility, and ANP32E is really just controlling H2AZ dosage. So the more H2AZ you have, the more you can attract the remodeler to position nucleosomes and open up chromatin, which then allows transcription factors and polymerase machinery to access the transcription start site and turn genes on. And really, if you get rid of ANP32E, it's releasing this valve and you get way more H2AZ spreading to all of these other sites in the genome. If you clamp this down tightly, then you restrict the amount of H2AZ you would have in the genome. Okay, so I've told you now a little bit about what we think H2AZ is doing at the molecular level. And now I'm going to come back to what we think is happening in the context of development. So circling back to what we found with zebrafish, we went through and looked more at those H2AZ sites which were increasing in the ANP32E mutants. And we compared them to some recent data that had been published looking at different histone modifications during a little bit later stages in development. And what we found was that H2AZ was increasing at sites that were classified as poised proximal enhancers, meaning these are not necessarily at the gene promoter, they might be at distal regulatory sequences. Again, just to remind you, this is when we got rid of ANP32E causing H2AZ expansion, right? So that's the H2AZ mutant, I'm sorry, the ANP32E mutant here where we measured H2AZ. We looked at motifs that were present at these sites to get an idea for what biological processes might be regulated. And we found that these 4 transcription factor motifs were the most abundant in these sites where H2AZ accumulated: FOXD, SOX10, PRDM1, and TFAP2. And for those of you who are not developmental biologists, these are the main transcription factors involved in neural crest specification. So what is neural crest and what is specification? Well, it turns out neural crest are these very specific cells that arise from the ectoderm during development, and they I don't want to say maintain, because they might even establish multipotency later on during development, but they're often thought of as non-embryonic stem cells that are maintained in the embryo. And induction of these cells occurs through this cascade of transcription factors, including PAX, TFAP2, SOXD, FOXD, SOX10, a lot of the factors that we were seeing enriched at the sites where H2AZ was enriched. So this led us to then hypothesize, well, maybe ANP32E loss disrupts these neural crest cells. So to investigate this, we took a published data set where they'd specifically done RNA-seq on neural crest cells at later stages in development. So just so you're aware, this is a 16-hour-old zebrafish, which almost looks like a zebrafish. And a lot of the neural crest lineages have begun developing and differentiating. And in this paper, they simply purified out those cells and performed RNA-seq. We asked, okay, if these are the neural crest specific genes that are on in these cells, are they on prematurely in our 4-hour-old embryos when ANP32E is lacking? So to investigate this, we performed gene set enrichment analysis. And for this, we take all of the genes that are turning on and put them on one side of the plot, the x-axis. All the genes that are turning off reside on the other side of the axis. And then we simply plot all of the neural crest specific genes at their respective locations along this x-axis. And based on a hypothesis, we would expect if ANP32E loss causes premature activation of these neural crest genes, we would expect them all to pile up on the on side rather than on the off side. And indeed, that's exactly what we saw. So it seems like ANP32E loss, which again causes H2AZ expansion, did indeed occur in a way that activated these neural crest specific genes. And we're not quite sure why the neural crest specific genes are activated. We have some hypotheses that we're currently investigating, but it does seem like H2AZ might have this very specific and unique function at neural crest cells, or I'm sorry, at neural crest genes, potentially in the neural crest lineage. To further investigate this, we've done some preliminary studies where we've now gotten rid of the H2AZ installation enzyme called SRCAP. To remind you, the idea here is that if we get rid of the SRCAP enzyme, then we'll lose H2AZ installation. Here we expected the opposite phenotype. Rather than causing premature activation, if H2AZ is involved in activation, we would expect failed activation of the neural crest lineage. So we performed these same experiments. In the SRCAP mutant, we found that there were defects in the jaw and in the heart, and in many other tissues and cell types that I really don't have time to go into right now. But when we looked specifically at cartilage in the jaw, we found that there was a major defect in cartilage formation. And the cartilage of the jaw in this particular region is derived from the neural crest lineage. And so this was a further indication that indeed H2AZ installation, potentially ANP32E regulation and SR-cap have this important function in specification of the neural crest lineage. And when we quantified this, we found that these cartilage in the anterior, I'm sorry, not in the anterior portion, but the posterior portion of the jaw were specifically disrupted when we got rid of the SR-cap gene. All right, so we've kind of settled on this kind of new idea for what H2AZ might be doing during development, and including the function of ANP32E. So what we think might happen is that at the neural crest genes, ANP32E might have a very specific function in preventing H2AZ from being installed. And this allows stem cells to be maintained as stem cells, where H2AZ is installed only at the genes involved in stem cell formation. But then as development progresses and neural crest specification occurs, ANP32E levels are actually decreased. And I didn't have time to talk about this, but we did confirm this looking at some recently published single cell data sets. And in this loss of ANP32E then allows the SR-cap complex to be relieved and install H2AZ at the neural crest specific genes. This then causes increased nucleosome remodeling and neural crest transcription factor binding, which then would drive the neural crest lineage. So in this way, H2AZ and transcription factors are kind of operating together at these sites in order to activate genes later on during development. Okay, so that's it. I'm going to end now. I will say that we've got some new studies that I haven't talked about where we think the same process is occurring during breast cancer progression, where H2AZ is expanding and some of these same neural crest transcription factors are allowing for epithelial to mesenchymal transition in breast cancer. If anyone's interested in that, we can kind of talk more about that, but it's some very new, interesting results that we have on this. Okay, so I want to thank the people in my lab who've done most of the work. Fan Ju is involved in both the mechanism question on how the remodeler acts at H2AZ sites, along with Kristen. Fan Ju has also taken the lead on the Zebrafish jaw project, and many others have helped us throughout this process, including my former advisor and some people who've helped us bring Zebrafish to the University of Rochester. And then I'd like to thank my funding sources. And so with that, I'll take any questions you have. And I really thank you for giving me the opportunity to present to you guys today.
All right, thank you. That was great. It's very informative. Questions?
I have a few questions. So what, in terms of what chromatin remodeler, would it be Swiss SNF, or would it be, what is it that you're expecting as being recruited to these marks?
Yeah, so we found that it was SMARCA4, which is Switch SNF. It's the bird1 flavor of Switch SNF.
Right. Yeah, so just a full disclosure, I study neuroblastoma, which comes from neural crest, and we've sort of honed in on ARID1A, and its loss leads to a mesenchymal phenotype. So I'm curious if sort of more the PBAF complex typically goes to these sites.
Excellent question. Yeah, so we haven't investigated that in that context, right? But it would certainly fit with our model, where the switch SNF complex, and which contains ARID1A, might be regulated in a manner H2AZ dependent potentially, but especially remodeling these particular sites would then allow for transcription factors to be activated, potentially driving epithelial cells to more of a mesenchymal phenotype. So it does, the results that you have certainly do jive with what we're thinking is going on. And I don't know why it would be these specific sites in the genome. I have a hypothesis that we haven't tested. Trying to write a grant on it, but
so don't talk about it in front of others. That would be wise.
Yeah.
So, and then is it just at specification? These are being found at it just at specification of neural crest, or is it during the first few stages of neural crest, including when they're migratory neural crest?
Another great question. So since I put this talk together, last month or so, and I've kind of been polishing it up, we have some new data that isn't quite ready for showtime, where we have measured H2AZ changes at various stages in development and in the neural crest lineage. And what we find is that H2AZ is actually expanding beyond its normal locations during neural crest development, even from the earliest stages all the way through migration and differentiation. And so I don't know why H2AZ has a specific function in the neural crest, but it does seem like it's happening throughout the process.
And because it seems important for fate choices, and obviously the neural crest is atypical because it allows both two whole different lineages, like mesenchymal, and so because it's kind of default is mesenchymal, and then it makes by basically fate choices to become neuronal or stay mesenchymal. So I'm wondering if it's just involved in the plasticity, and so I kind of missed, unfortunately, the very beginning of your talk, but this is also a mark that's prevalent. Obviously, I believe I was seeing in basically in sperm and other organs where you'd want to maintain as many lineage fate choices as possible. So is it because it's related to lineage fate choice? Like, is it very fundamental?
Have you read my grant?
I have not, no.
No, I'm joking. No, this is exactly our hypothesis, right? So in the stem cell, we find that these H2AZ sites are the platform for the establishment of bivalent chromatin.
Right.
And as you point out, during neural crest development, there's this maintenance of multipotency. And so what we think might be happening is that H2AZ is expanding from its prior locations to new locations so that it can establish bivalent chromatin at new sites, enabling multipotency of the neural crest. That's our basic idea.
Right. Yeah, I mean, that makes sense to me because neural crest is such an unusual cell population. It's just it's recapitulating something that's foundational in terms of development. So yeah, very, very interesting talk. Thank you.
Yeah, thank you for your questions. They're perfect.
Thank you.
I'll ask a question if you don't mind. So would you assume that a SMARCA4 inhibitor would mimic the phenotype that you're seeing?
That's a great question. I guess so. Yeah, I mean, it would have to be, it would have to be a carefully controlled experiment, because what we're trying to do is not completely destroy SMARCA4 function. We're just trying to, diminish function a little bit. So if you applied a dosage curve or something to a SMARCA4 inhibitor, we would expect that it should be able to recapitulate that phenotype. It's a great idea. We haven't done that in zebrafish. I'm actually not aware of a SMARCA4 inhibitor that would allow us to do these experiments. So if you know of one, I'd love to hear about it. Yeah, it's a great idea.
I shot you an email and a note in the chat because we have a bromodomain inhibitor of SMARCA4 that we're working on. And I think that could be a really cool collaboration.
Yeah, that would be awesome. I would love to have a SMARCA4 inhibitor.
Okay, well, I shot you an email. Please follow it up when you get a chance. But it was great talk. Really enjoyed it.
Thank you so much.
Any more questions? Graduate students, feel free to chime in. I guess I have a really, really general question. So what is kind of, so I'm thinking about so open chromatin in general, and a lot of people will use like gene expression profiles to classify cells. But I'm wondering about open chromatin profiles in terms of classifying cell type, and sort of what is kind of the most simple classifier of cell type. Is it like open chromatin? Is it open chromatin plus whatever factors might be bound there, or is it so what? I don't know. I was just kind of thinking about that during the talk.
No, that's a great question. I really like this question a lot. So, there's a lot of different characteristics of a cell that you can use to classify what cell type that is. And recently, people have started doing these single cell RNA-seq experiments to cluster cells and define their potential tissue type and so on. Similar studies have been done on chromatin accessibility, and you generally get the same result where chromatin accessibility can be used to classify cell types. We've actually, so some of the stuff I was hinting at in the last slide, we've actually gone into the TCGA project, the Cancer Genome Atlas, and we've downloaded hundreds of chromatin accessibility data sets in an attempt to define breast cancer and other cancer cell types, specifically getting at the question that you're asking. And it turns out that if you use some of these machine learning based techniques to classify chromatin accessibility data, you can in some ways get almost identical clustering that you would get from gene expression analyses, but it's not identical. There are certain differences. And so what we're finding is that, for basal-like breast cancer, triple negative breast cancer, for example, chromatin accessibility and gene expression are really tightly correlated. Similar for luminal A or lobular breast cancer. But then when we start looking at more hormone positive breast cancer, it seems like there's a lot of subtypes where gene expression patterns do not specifically match the chromatin accessibility differences. And so we're thinking we might be able to learn something new about how hormone responsive or hormone positive breast cancer might be regulated at the chromatin level. And actually, we're studying how ANP32E might be involved in that process as well. So I guess that was a long answer to your question, but the short answer is it just tells us something different, right? And I don't know if it's necessarily better or worse than gene expression, because I think we have to think about cellular programming in the context of all the things that might be happening in a cell.
I just have a quick question. This is also more of a theoretical question. This is not quite my area of study, so I'm sorry if it's a little simplistic, but is this more of a kind of thing that's being used for things that are sort of one-way streets? Like in development, things are kind of a one-way street. You stop expressing something or you start expressing something as a cell develops. But there's a lot of genes where there's a lot of fluctuation. Do you think this mechanism is involved at all in genes that have fluctuating patterns of expression?
Yeah, absolutely. Another great question. So I like to think about epigenetic marks in general as controlling stochasticity, right? And so if you have really stable epigenetic marks at regulatory regions, then this would kind of define your classic developmental cascade, right? But if there is an epigenetic mark or pattern of epigenetic marks that might be a little bit more dynamic or less stable, they might underlie stochasticity in cellular programming and potentially allowing cells to transition back and forth between states or gene expression patterns to fluctuate. So I think this is exactly what we're thinking about. So great question. Thank you.
Do you have any idea of timeframes that some of this stuff could happen? Because there's things that are like circadian related gene expression. Is any of this involved, or is that too narrow a time window?
It's another good question. I don't have a real answer for you, but I can imagine and hand wave. I know that the nucleosomes where variants are installed occur outside the cell cycle. So unlike the canonical nucleosomes, which are installed as the cell divides, these nucleosomes can be installed at any time. So if there were a epigenetic mark that would allow for more short time scale transient changes in the genome, these histone variants like H2AZ would certainly be my top pick. So I guess the answer is I don't know, but it could be these marks.
Thank you.
You're welcome.
I just wanted to ask a little bit more about the breast cancer point that you made, that you see a broader sort of epigenetic repertoire versus sort of gene expression. I mean, it may relate to the just previous question, is it just possible, were those studies done with, single cell seq? Is it possible that you have in that population expression at a low level and some of those open sites that are not showing up in the sort of the averages that you get in bulk sequencing or RNA sequencing?
Absolutely, yes. So, a lot of times the classification occurs through histology, and there might be a mixture of cells. Some cells might be expressing a particular marker and other cells might not be. And really it's up to the clinician or the histologist or the pathologist to go in and binary decide, is it this or is it that? Right. And biology is not that simple. So what we might be classifying is certainly not including the variation that exists within these tumors. And that really might be what we're uncovering with the variation in epigenetic marks as opposed to the gene expression changes or differences, I should say.
Yeah, and I would just sort of say that it might be interesting because some of what we're interested in is how does cancer hide from therapy or adapt around cell identity switching? So effectively, I'd be kind of curious if something had a broader sort of epigenetic profile versus expression profile, whether that would be associated with worse outcome, whether that might predict poor outcome.
So actually, we do find that I guess what I should say is I have to be careful about the way that I say this because we've been analyzing things in so many different ways. What I will say is that the more broad epigenetic profile, if we're calling it that, is certainly associated with more advanced stages of cancer.
I see. Yeah.
So it might not necessarily at this point associate with outcome because we're sampling patients when they come into the clinic, but if we were to do longitudinal studies, we might be able to get at that. We just haven't done those kinds of studies.
Right. All right. Well, thank you very much. Enjoyed it.
Yeah, thank you.
Hi, Dr. Murphy. Great talk. I really enjoyed it.
Thank you.
I was curious, actually, you mentioned development and you also mentioned cancer progression, but I was curious to see if you knew of any other processes that this H2AZ localization takes place in.
Sure. So, so this collaboration that I was talking with the students earlier, for those of you who weren't there, I'm now working on learning and memory in mouse, and through this interesting collaboration with colleagues at the University of Toronto. It turns out H2AZ is removed from many genes and enhancers in response to a stimulus triggering memory formation in neurons. And H2AZ removal is necessary for that process. If you prevent removal, then it doesn't happen. And if you prematurely remove H2AZ, then the mice fail to remember things. And so we actually find that you can increase the ability of the mouse to remember things if you muck around with H2AZ at particular sites in the genome. So it's again, the same molecular process, we think, where H2AZ is installed. It allows for kind of reprogramming of gene transcription networks. and then it can kind of stabilize and allow a new program. So it's just a different cell type, right? It's the same molecular process operating in a new cell type. And I think the theme here is plasticity, right? We want to maintain a cellular program that is responsive to change, but then can be stabilized afterwards. That's what we see in the stem cell. That's what we see in the neural crest cell. That's what we see in the breast cancer cells. And then again here, that's what we see with the neurons. Great question. Thank you.
Thanks for the answer.
Any last questions? All right, well, I think this was a fabulous talk and really great questions, and I really enjoyed the discussion. And thank you for your time. Chelsea, thank you for inviting Dr. Murphy. And if there's no more questions, thanks everyone, and I would say have a great weekend.
Thank you. Thanks everyone.